about the work that we are doing in the lab. The lab shares with you, I guess, a great fascination for this thing that's displayed here, the neuron, and what it's all doing, how it's possible that integrates so much information and gives rise to many of the functions of the brain. Of course, I mean, you're all aware that this extensive dendritic tree integrates information from many other cell types, and that's what I wanted to talk to you about how this integration is brought about, and in what way this integration is altered by neuromodulation. But in the lab, we have also a very strong drive to let's say, to probe the relevance of mechanisms that we figure out in preparations, the relevance for animal behavior, or the relevance for the human brain. So that's why I know that you heard Guillermo's talk yesterday. We have a very nice collaboration now ongoing with neurosurgeon in Amsterdam, and try to test these open questions of how are these mechanisms that we figure out in the rodent brain most of the times, how are they relevant for the human brain? Okay, so what I will do is I will show you a couple of stories, little parts of stories, in which I will tell you about dendritic integration, modulation of dendritic integration, and plasticity, whether it can play a role in animal behavior, and what these mechanisms have to do with the human brain. So, of course, these the cortical networks, the cortical pyramidal neuron integrates, as I said, a lot of information from incoming from other neurons. But these networks themselves are again under control of neuromodulation, and one of the prominent neuromodulators is acetylcholine. So the cholinergic system in the brain, basal forebrain neurons project to all cortical areas and release acetylcholine there, and especially when we are awake, because the traditional view on how acetylcholinergic signaling in the cortex, the role it plays, is that it during the day cholinergic levels are elevated, and they allow us to focus, to be aroused, to be focusing on our attention on incoming sensory information, but that this type of signaling is actually a very slow type of signaling. That's the traditional view of cholinergic signaling in the brain. However, this view has recently been challenged by fast cholinergic measurements using electrochemical detection probes, just like with dopamine detection in the striatum, for instance. That people have discovered that cholinergic signaling also is actually quite rapid, happens on the scale of seconds or even faster, and it is very much linked to animal behavior. So in this example, the cholinergic detection probe is lowered into the prefrontal cortex of a rodent, of a rat. Of course, the prefrontal cortex is crucial in focusing attention. So that's what the experimenters did. They showed the animal a cue, and when the animal successfully detected this cue, then the acetylcholine levels in the prefrontal cortex rise rapidly when the cue was detected. But when the cue was missed, animal was not paying attention, then acetylcholine levels also did not show any changes. So apparently these changes in acetylcholine are very much linked to cognitive operations, and especially Martin Sartre really believes that.
What is the causal relation?
Sorry? Well, the causal relation, very good point. So we will get to the causal relation later on in the talk. So here the correlations in this Martin Sartre's lab, a lot of correlations have been revealed about this. Of course, he's now also probing this with optogenetics, so that he can really make a causal role. But I will tell you some more about causal links between cholinergic signaling in the brain and attention behavior in particular. Anyway, so what kind of receptors do cortical neurons express to sense acetylcholine levels? So of course there are muscarinic receptors. They are G-protein coupled receptors, are very well known for network synchronization. So many of the EEG studies show, of course, that the brain, that the cortex shows synchronous activity at different frequency bands, and acetylcholine plays a very significant role in synchronizing activity in cortical networks. I will not talk about muscarinic receptors, but the other type of receptors are nicotinic receptors, and they are ligand-gated ion channels. There are different types in the brain, and they conduct fast currents in cortical neurons. And these currents, they appear at the time scale that this fast cholinergic signaling is occurring. So what I want to do in this talk is to talk about the prefrontal cortex, nicotinic receptor expression in the prefrontal cortex, and how neurons in the prefrontal cortex are modulated by nicotinic receptors. So the prefrontal cortex in the rodent consists of several layers, of course, like other cortical areas. What it is different in is that it doesn't have a layer 4, a classical input layer. It lacks. So instead of thalamic inputs arriving into the prefrontal cortex in layer 5 and layer 6, and hippocampal inputs arrive in the prefrontal cortex through layer 1 and 2. And of course, the prefrontal cortex is connected to many other, almost all other brain areas. So of course, when you think about information processing in the prefrontal cortex, information from other cortical areas comes in layer 2, 3. It's processed there, of course. The output layers of the prefrontal cortex are still layer 5 and 6. So layer 5 projects to striatum, for instance, and layer 6 and amygdala. Layer 6 projects to back to the thalamus and other cortical areas. So, so these are the output layers. So the questions that I want to address in this talk is: how does nicotinic receptor activation alter prefrontal cortex network function? How the activation of these receptors affect the rules for spike timing and plasticity? Do nicotinic receptors in the prefrontal cortex play a role in PFC-based behavior, so attention behavior mainly? And then at the end I will tell you something about how whether human synapses, what kind of spike timing dependent plasticity rules they show. Okay, so coming back to the first question, how does nicotinic receptor activation alter the cortical neuronal network function? So the first approach that we took was to just look at the network as a whole of the prefrontal cortex, make sliders of the prefrontal cortex of mice, and then load the sliders with calcium dyes and look at acetylcholine-induced activity in these slices. So it looks like this. If it wants to play, all right. So what you see is that the neurons are loaded, and every now and then you see neurons that are activated, lighting up by increasing fluorescence when action potentials are fired. So of course you know that these changes in calcium are related to action potential firing. When we analyze the changes in fluorescence in these individual neurons, then we see that when acetylcholine, especially when acetylcholine is present, there's an increase in these fast signals that are related to action potential firing. And we can do that for many neurons at the same time. And then when we analyze the different layers or the different depths in the prefrontal cortex, what we see is that especially the deep layers show much more activation in response to acetylcholine application than superficial layers. So of course when we do two-photon imaging of networks, one big advantage is that you can go back to the neurons and zoom in onto the neurons and look at the identity of neurons. So what we did is we made high-resolution pictures of these neurons and tried to determine whether activated neurons were either pyramidal or non-pyramidal cells. And what we find is that there's a difference in the activation between pyramidal and interneurons, and that especially in the superficial layers, it's not so much the pyramidal neurons that are activated by acetylcholine, but it's more the interneurons that are activated by acetylcholine. In other layers, the deeper layers, especially layer 6, the pyramidal neurons show much more activation and a certain amount of interneurons. So apparently there's a certain kind of distribution of nicotinic receptors that gives rise to this difference in activation of different types of cortical neurons. So
did you say before that the deeper layer are more activated?
Yes,
but how do we see it from here?
So these
are the deeper layer to the right, no?
Right.
It's
So
only I understand that it's the ratio, but
right, so the percentage of neurons that were activated, it's sort of ratio indeed. So about here it's about half pyramidal neurons, half interneurons that are activated. But here it's predominantly interneurons in the superficial layer. In this picture you can see that the cells, the activated or the percentage of cells that are activated is higher in deep layers than in superficial layers.
But what about the concentration of interneurons versus pyramidal?
In the different layers?
Yeah.
Ah, okay. So if I, let's say, rephrase that would mean that is it possible that in superficial layers there are more pyramidal neurons or less pyramidal neurons, more interneurons?
Yeah.
Right. I actually don't know.
Do you mean by interneuron between layers?
I mean by interneurons, non-pyramidal neurons. Non-pyramidal neurons, so neurons that don't have a pyramidal shape.
So let me understand, how do you stimulate? What has been activated?
So we don't stimulate, I mean, the only thing we do is we image the slice, it's loaded with calcium dyes, we just do nothing, image the activity, then before, and then the only thing we do is we apply acetylcholine.
no, they apply acetylcholine. But it's not
Apply acetylcholine.
stimulation is the
That's
application.
okay.
So it's not electrical stimulation, it's
I
just
understand.
applying.
You apply acetylcholine,
Right.
and then you measure the activity.
Right, we measure, we just count these
This is fine. Okay.
blips.
But you do it before and after?
Yeah, before, after,
No,
before, during and after.
But those histograms that you showed,
Right.
what has been activated? More than without the acetylcholine or just?
So what you do is you count the number of neurons,
Yeah.
and then what's displayed here is the percentage of those neurons that show a blip during this time.
So here this is the baseline.
Yeah, the
Right.
green
Green is baseline.
I know, but in the next histogram, the next slide,
Ah, okay.
the
So
slide here.
what we do is you take the neurons that are activated by acetylcholine.
What does it mean? Maybe the same percentage is also without acetylcholine.
So the difference between these two.
We take
So
the difference.
So I mean, we follow the fate of the neuron. So we can know of the whether the neuron is going to be responding to acetylcholine or not, whether it shows more of these blips during acetylcholine than before. So that would be called an activated neuron, a neuron that's activated by acetylcholine.
So
And
you
then,
look at the blue neuron, you look at those that are activated by the acetylcholine, and on those you are trying to see whether they are
right.
inhibitory.
Do they show the typical pyramidal shape that you can show? You see here, or do they show some other bipolar or multipolar shape? And then we call them non-pyramidal or interneural. So
Anyway,
this
I think it's true to say that in most cortices, the percentage of inhibition is about, let's say, 20%.
right. Yeah.
In all layers.
Right.
Some layers are a little bit
Right.
more excitatory, but in general it's 20 to 80,
Right.
basically.
Yeah.
So this keeps
Yeah.
true for all layers.
Right. So the conclusion that we draw from this is that most likely in superficial layers, pyramidal neurons are not so much activated, but it's interneural. So of course all these questions are very valid. So to really know what's going on, we went in, we targeted these neurons with recording electrodes and just started recording the different layers and characterizing where are these nicotinic receptors. So when you record from pyramidal neurons in layer 2-3, so we go through the circuitry layer by layer. When we record from pyramidal neurons in layer 2-3, then we see that most of the neurons don't show an inward current, since these nicotinic receptors give rise to an inward current when we apply acetylcholine. So the majority of neurons doesn't show a response, whereas in interneurons you see clear responses, either a very fast current that's carried by an alpha-7 type of nicotinic receptor. We confirmed that with knockouts in pharmacology, or a slower response that's carried by a different type of nicotinic receptor, alpha-4 beta-2 type of receptors. So in layer 2-3, what you indeed see is that in pyramidal neurons there's not so much nicotinic receptor expression, but it's more in the interneurons, and also excitatory inputs to pyramidal neurons don't show nicotinic receptor modulation. Layer 5 neurons are different because they express this fast type of nicotinic receptor. About half of the cells, or a little bit more than half of the cells, express these currents. Interneurons, again, we probe different types of interneurons. fast spiking cells in half of the cases show these fast currents also, aldosterone, and the regular spiking non-pyramidal and the low threshold spikers, they both show slow inward currents, and the LTS cells show a combination of the two, so a fast inward current followed by the slow inward current. So in layer 5, we get a different picture. Also, the thalamic input to layer 5 neurons show nicotinic receptors. I didn't show the data, but that's so this layer 5 is much more strongly regulated by nicotinic receptors, both pyramidal neurons and interneurons. Layer 6 pyramidal cells yet again show a different type of response to acetylcholine application. They show a slower response, which is carried by beta 2 containing receptors, nicotinic receptors. So this kind of current, this kind of inward current, will give a very prominent depolarization to a neuron. Of course, you can do pharmacology and you can record knockouts of beta 2 receptors, and that shows the typical signature of the alpha 4 beta 2 receptors. So that's in most of the cells that show the majority of the cells shows this the expression of this type of receptor. So summing that up for layer 6. Pyramidal neurons show alpha 4, beta 2 receptors. Interneurons also have these receptors.
I'm curious about that. What do you mean by modulating the network? I mean, it's just you activate those receptors and you stimulate the cell.
Right.
So why this is modulating the circuitry? In what sense it's modulating the circuitry?
So in the sense that it changes the activity of the cell. So if acetylcholine,
because you have another drive to the cell, but it's not.
Sure, of course, yeah.
It doesn't change the circuitry.
So not all cells are equal, apparently. So not all cells express the same nicotinic receptors to the same amount. So superficial layer pyramidal neurons don't express nicotinic receptors.
I understand that, but when you say modulate the circuitry, I have a picture where having activating those receptors modify other synaptic inputs, modify the circuitry.
Right. So
but
synaptic
you are not
inputs.
showing coupling between the excitatory and the synaptic, the ionic synaptic currents.
Sure, I didn't show the data, but I showed the receptors here. So inhibitory inputs to these cells, they get modulated because the neurons get activated strongly. The neurons get activated strongly. Excitatory inputs get modulated in layer 5, but not in layer 6, not in layer 2, 3. But I don't show this data because I want to show much more other data.
Okay.
I just want to give you a picture of you have to skew the cortical circuitry as expressing, differentially expressing nicotinic receptors. So it depends on the cell type and on the layer what kind of receptors they will express and how they will be modulated by nicotinic receptor activation. Okay, so that's the point here. But of course, I mean, this is a very basic characterization, boring stuff. Of course, what you want to know, of course, what is it doing to the function of the network? Of course, so when we have identified all the locations of these receptors, how is that actually the function of the network altered, and in particular, how is synaptic, for the first question, how is synaptic plasticity affected by activation? Because you can imagine that if you have presynaptic and postsynaptic receptors that are ionic, that give depolarizing ionic currents, it's hard to predict how synaptic plasticity is going to be affected. And there's also, of course, if you have, since these plasticity plays a role on synapses along the dendrite, if interneurons are going to be activated along this dendrite, they will by acetylcholine. Then in the presence of acetylcholine, this dendrite might even suffer more inhibition than activation.
So is the amount of ACH that you pull there in the plastic relevant to what's secreted?
To synaptic ACH. So it's a pretty high concentration, but it's at the, let's say, the far end of the dose response curve for the receptors, and it's also what is found in synapses. Another question, I think, somewhere. Okay. So we looked at spike time independent plasticity. Of course, you all know what spike time independent plasticity is. Just to shortly refresh your memory, spike time dependent plasticity is plasticity that depends on the precise timing and the order of activity of pre and post synaptic neuron. of course, when the classical view of it is that when a presynaptic neuron is activated first and a short amount of time after that the postsynaptic neuron fires, there's an increase in synapse strength. If you reverse the order, postsynaptic neuron fires first and the presynaptic neuron fires shortly after that, then you get a decrease in synapse strength. And of course, the classical, by now classical picture of hippocampal from hippocampal cultures is that in this small window where you have this order of activity, you get a decrease in synapse strength, whereas in this case you get in a small window of time you get an increase in synapse strength. Okay, so the question is, what is acetylcholine or nicotinic receptor activation doing to this phenomenon, and what are the mechanisms underlying that? So we recorded in layer 5 of prefrontal cortex and induced spike-time dependent plasticity, having a very short window between pre and postsynaptic activity, and then you get a sustained increase in synapse strength. When you apply a very small concentration of an agonist of the acetylcholine nicotinic receptor, nicotine itself, then you see that this increase, you do the same protocol, is now changed into a slight decrease of the synapse strength, and you can see that this is a different concentration is occurring, and when you block the beta 2 containing receptors, you block this effect of nicotine, so it's involving nicotinic receptors. This result was published a couple of years ago, but what are the mechanisms underlying this effect of nicotine? I've shown you now that nicotinic receptors are distributed among the neurons in the prefrontal cortex, but how can you have an excitatory ionotropic receptor that gives rise to this decrease in plasticity? So what are the mechanisms involved? So first of all, of course, also in the prefrontal cortex, spike-time dependent plasticity requires changes in calcium postsynaptically. So when you do the same protocol, but in the recording pipette you include a calcium chelator like BAPTA, you don't get increase in synapse strength. So what we did is we imaged calcium in the dendrite of layer 5 neurons and looked at changes in fluorescence in response to action potential firing. And what we saw is that in the presence of nicotine, actually this change in calcium, dendritic calcium in these neurons is much smaller than, smaller, is reduced with respect to control conditions. So if nicotine receptors are excitatory and they conduct sodium and calcium, how can nicotinic receptor activation reduce calcium signaling? So one of the likely candidates, of course, is that interneurons are activated by nicotinic receptor stimulation, and that's what we tested. So if we looked at, we stimulated inhibitory inputs, 2 pyramidal neurons, and then applied the agonist of acetylcholine receptors, nicotine. You see that there's an increase in the amplitude. So this is, of course, the result of the fact that nicotinic receptors are expressed by interneurons. So the synaptic, so in the presence of nicotine, the inhibitory synaptic synapse increases in amplitude and decreases after nicotine again. Now, so to test the hypothesis that this increase in inhibition is the mechanism by which nicotine reduces spike-time dependent plasticity in these layer 5 neurons, we blocked GABAergic receptors with gabazine. We did the same experiment again, applying nicotine when we ran the spike-time dependent plasticity protocol, and then you see that there's no longer a reduction in plasticity, but there's still an increase in synapse strength.
Let me try to arrange it. So you stimulate extracellularly.
Right.
You activate many cells.
Mm-hmm. Sure.
So this spike-time dependent plasticity is not for one particular type of synapse, but it's a combination. You record a combination of plasticity, so to speak, for many cells, and you claim that some of it is due to the effect of inhibition on this spike-time. It's not per synapse, it's per population.
Right, sure.
So because in the population there is inhibition,
right.
In this population you increase inhibition due to nicotine, then you reduce effectively the spike-time independent plasticity. But it's not that the synaptic plasticities per connection is changing, but the population is changing.
Right. So
if I understand correctly.
Right, no, you understand, you described correctly. So what we think is that indeed, when we stimulate extracellularly, you would stimulate both excitation and inhibition. When you apply nicotine, different kinds of interneurons, among which these low threshold spiking cells that project to the apical dendrite of these pyramidal neurons will be activated more. The amplitude will increase, thereby impose more inhibition on the dendrite, and thereby effectively reducing this calcium signal in the apical dendrite, which is necessary for the change in synapse strength.
But if I take two pyramidal cells, suppose you were doing peri-coatings from two pyramidal cells, with nicotine and without nicotine, then this explanation will not hold. This particular synapse that is not involved with inhibition should not be affected by nicotine itself, if I understand the mechanism.
Only if the inhibition on the dendrite is somehow timed with that.
No, there is no inhibition between two
No, but
pyramidal
then
cells.
in addition, there are on the same dendrite inhibitory. In addition.
But I don't activate them when I do peri-coating.
No, but
Right, so
simultaneously activate them.
I can simultaneously.
Then you have this effect.
Of course, if you have an interneuron in between.
Oh, you mean the inhibition affects this peri-coating because then it reduces the calcium which is needed for this particular. Okay, I understand.
So the backpropagating action potential is most likely affected by inhibition imposing on this dendrite. And if you increase the inhibition, that's the argument. Okay, so of course, so the conclusion then basically is that distributed network effects of nicotine, by which I mean that nicotinic receptors are distributed in a particular way across the network, gives rise to a change in rules for synaptic plasticity when acetylcholine or when nicotinic receptors are activated. So if you think of this fast cholinergic signaling during attention, then this is what may happen, that during those moments of attention, the rules for synaptic plasticity may actually be different than in the absence of acetylcholine. Of course, the relevant question is, at least in my mind.
But then you learn less with attention. That is what you learn. When you are attentive, you learn less.
So, of course, so we didn't scan the whole plasticity time window in these neurons. So how the whole curve would change, I actually wouldn't know. At this point, we didn't test that. But and then of course there's the other point that of course it's very tempting to say increase in synapse strength is learning something, decrease in synapse strength is forgetting something. That's a very shortcut.
But this is not changing. Not just this is not changing.
So the only thing that I think the only thing we can conclude from this is that in the presence of acetylcholine, the rules for synaptic plasticity may be different because the network is in a different activity state. Since different neurons are affected differentially by nicotinic receptors, it is relevant to figure this out.
But did you look at the LTD part of the
exactly? So we didn't scan for the whole curve, and we didn't look for the LTD part, but that's would be an important next step. So, are these nicotinic receptors in the prefrontal cortex? Do they play a role in prefrontal cortex-based behavior? Of course, I showed already that there is correlation. That during attention, during detecting a cue, there's fast changes in acetylcholine levels. So you might expect that the receptors are relevant during that behavior. But as was pointed out earlier, these are all based on correlations. So based on correlative observations that during attention there's some change in acetylcholine. But we of course want to have a much more rigorous answer whether these receptors are involved. So what we did is we addressed this by looking at attention behavior in rodents. So there's a particular task which is called the 5-choice serial reaction time task, which is developed in Cambridge by Trevor Robbins and his colleagues. And different domains of attention. The task looks as follows. So what you're looking on top of an operant chamber in which a rodent is sitting. And what you see here is in this side of the chamber there are 5 holes, and at this end of the chamber is 1 hole in which food reward will be delivered. So the animal has to pay attention to these 5 holes, and in one of these holes, for a very short amount of time, at some point the light will come on. So the animal has to bring its nose into this hole, and then if it does that so correctly, within a certain amount of time, then it will receive a food reward at this end. So all the animals are a little bit hungry when they enter into this chamber, so they're motivated to do this, and they can learn this pretty well. So for simplicity's sake, you can score many aspects of this behavior, but for simplicity's sake, I will talk about two parameters. The first one is the percentage of omissions, so the omitted, the failure to respond. So you can view this as the animal is doing something else, it's not paying attention to this board, it missed the light completely and it does not show a response, would be an omission. And then the percentage of accuracy, so it's the percentage of correct responses. So what could happen is that the animal pays attention, the light comes on, but it sticks its nose in the wrong hole. That would be scored with this parameter. Okay, so first of all, what we did is we looked at mice that lacked the beta-2 receptor, beta-2 containing nicotinic receptor completely. So the beta-2 receptors were these slow currents that are expressed in pyramidal neurons in deep layers and by many interneurons across the layers. It's a very abundant subunit present in the animal. So in this case, it's a very blunt experiment in the sense that beta-2 receptors are lacking everywhere in the body. So even in the tail or in the left foot of the mouse, but also in the brain, the beta-2 receptor is not there. Also in the prefrontal cortex. So what you see is that the accuracy, when we look at this parameter, so the number of correct responses is very similar in these wild type and beta-2 lacking mice, but the number of omissions is is elevated in mice that lack beta-2 nicotinic receptors. So apparently somehow these mice don't pay attention so much. During the whole session, during the whole training, they are very motivated to do it. So they eat the same amount in the end. They show the same latency to collect the reward and stuff like that. So they show the same locomotor behavior. So on those basic parameters, they seem to not differ, but they somehow pay less attention. So this would say, well, you need these beta-2 receptors to be expressed to show proper attention performance. So this is done at when the stimulus light is on for 1 second. So it has to detect that light during that second to be able to make a response. But you can increase the attentional load by reducing the amount of time that this light is on. So you can reduce the amount of time that the stimulus is on to a quarter of a second. And what we see is that in all animal groups, the number of missions goes up and the accuracy goes down because it's more difficult for the animal to do this, to pay that amount of attention to do this. But what you see is that for all stimulus durations, these knockout animals perform worse than the wild types, show more omissions, show less attention. So of course, what we want to know is, of course, this is only showing.
Would you expect that?
Would you expect that? Well, of course, if acetylcholine signaling is relevant for attention behavior, then lacking an important receptor, an acetylcholinergic receptor, would interfere with attention behavior. Sure. So in that sense, it's a very simple prediction. Sure. So a lot of pharmacology has been done and has shown the same thing, but it's all, of course, very. Pharmacology is always systemic. It's never very into the PhD. It's a very different time domain.
But with these original mice, is this the Maria Picciotto mouse?
Yeah,
I mean,
this is Picciotto mouse.
They showed a change in the avoidance of dark. Would you attribute that to the lower attention?
Because they don't want to look at the light, that's what you're saying? So the light is, of course, on for 1 second. You never know. I mean, maybe they have a sensory problem in that sense. To us, there's no indication that that's the problem. Okay. But of course, I mean, it's very relevant or very just question, and of course, we wanted to address that. So what we did is these kind of experiments show that most likely you need this receptor. It's necessary to do this experiment, but of course, what you want, the receptor is missing everywhere in the body, in the brain, Of course, what we want to know is the beta-2 receptor. When you put it now back in the only in the medial prefrontal cortex, so re-express the beta-2 subunits in the medial prefrontal cortex only with lentiviruses and DNA containing DNA for its subunit, would you then get a functional restoration of this response? It would also take care of that point that you raised.
That'd be interesting. What would affect the slope of that line?
We will see. It won't affect the slope of the line, but I will show you what it will show. So what we did is we used a tool that was developed by Huib Mansvelder in Paris, where the mouse was also made. It's a lentivirus that contains either EGFP or EGFP together with the beta-2 DNA for the beta-2 subunit. And we injected this lentivirus into the prefrontal cortex of different types of mice, wild-type mice, beta-2 knockout mice, or beta-2 knockout mice. And in 2 groups, we only injected the control virus containing EGFP, and in 1 group, we injected this virus containing the beta-2 receptor. This to see whether the beta-2 receptor there would be sufficient to restore attention performance. But of course, first we needed to see whether it was actually expressed in the medial prefrontal cortex. So what we did is some immunocytochemistry against EGFP, which shows that neurons are transfected with these lentiviruses and start expressing EGFP. When you do a new N staining for neurons, then you show that there are many neurons that show an overlap between either EGFP and new N, the yellow neurons. But of course, as you saw in the vibe, this does not show that these neurons contain beta-2 receptors, and also beta-2 receptors by themselves cannot make nicotinic receptors. So the neurons need to express other subunits, alpha subunits, also to make functional nicotinic receptors. So you don't know whether these neurons, these yellow neurons, actually express nicotinic receptors. So to do that, we started patching from EGFP positive neurons and applied acetylcholine to see if their neurons have functional receptors. So that's shown here. So an Alexa loaded pipette is put onto, making a whole cell recording from this EGFP positive neuron. And then we tested in the 3 groups where we saw nicotinic receptor responses. So in the wild type neurons, of course, you expect responses because I've showed them already to you in other wild type animals. In the knockout neurons, you don't expect beta-2 responses, and you don't see beta-2 responses. But then when we have the knockout animals that have beta-2 receptors injected into the PFC, the DNA for that, then we see functional restoration of beta-2 responses that can be blocked by proper pharmacology. And the amplitudes are very similar between wild type and the knockout animals that receive the vector in the medial prefrontal cortex. So that shows that you can put back nicotinic receptors into the prefrontal cortex neurons and get functional expression. So is the re-expression, of course now we get to the point, is the re-expression sufficient to restore attentional performance? So we have here the 3 groups. Before injection, you see that the wild type groups, the knockout groups, show more omissions relative to the wild type group. And now we start to inject these viruses. And then what we see is that only in the group that receives the beta-2 containing DNA, we see that the attention level is back to normal again. So, and in accuracy, you don't see a change at all like before. So apparently, injecting viruses into the PFC does not harm their attention performance. So with functional restoration of beta-2 subunits in the medial prefrontal cortex restores attention performance in these animals. So that shows that these nicotinic receptors in the prefrontal cortex are necessary for proper attention performance, but also when you put them back, it's sufficient to put them back in the medial prefrontal cortex and not to get functional restoration of attention performance. And when you now change the stimulus duration and you look in the 3 groups, then you see that the wild type and the knockout are far apart, as I showed you already, and that the group that got the beta-2 subunit back into the prefrontal cortex, medial prefrontal cortex, is now lying close to the wild type group. I don't see a real change in the slope of that line, but at least the omissions are reduced again. And the accuracy follows the same pattern as I showed you before. So this shows that the nicotinic receptors, the beta-2 containing receptors in the prefrontal cortex are actually necessary for the behavior and sufficient in the medial prefrontal cortex to get the attention behavior going. Which points out to me that there's a causal relation between these receptors and behavior. Okay, of course, when you talk about nicotinic receptors, you can hardly avoid talking about nicotine and the nicotine that we that some of us like to self-administer, like our famous Dutch painter. When you follow the nicotinic concentration in the blood of smokers, you see that it is shortly after lighting up. In smokers, by the way, concentrated nicotine in the blood is never zero. It's always around 40 nanomolar. When a cigarette is lit up, then the concentration in the blood rapidly rises towards a half a micromolar. And then
one cigarette?
This is one cigarette. We follow one cigarette. So 3 minutes it starts to rise, then probably the cigarette is done. And then the concentration falls off.
Maybe it's good for the brain.
Maybe I should start.
Attention.
I should start to smoke.
And when you do it in patients in a PET scanner, when there's a PET ligand for nicotinic receptors, and the patient starts to smoke, what you see here is 0.0 cigarette. Then you see that there's no difference. 0.1 cigarette, then you see that already nicotine starts to drive off the PET ligand from the nicotinic receptor. 0.3 cigarette, you see that the whole brain is flooded with nicotine. 1 cigarette, a similar picture. And after 3 cigarettes, you get a little bit more occupation of nicotinic receptors, but not much than after 1 cigarette. So the whole brain is flooded with nicotine, also nicotinic receptors in the prefrontal cortex. But the question that I would like to address is, of course, the presence of agonists. Of course, if you know a little bit something about the receptors, always gives rise to desensitization. You cannot stimulate something forever without getting desensitization, and especially nicotinic receptors tend to do this to desensitize. So we wanted to know, well, how is desensitization? So if somebody smokes, what does this do to the nicotinic receptor functionality in the prefrontal cortex? So you can look at the different types of receptors again. So report from neurons, so there are 6 pyramidal neurons, for instance, that contain these alpha-4 beta-2 receptors. We apply acetylcholine a couple of times, and then start watching the smoker's concentration of nicotine for 10 minutes. And then what you see is that there's a gradual decline in the amplitude of the response to acetylcholine. And then that recovers over 45 minutes or so after this protocol, after nicotine.
So the attention is less?
So we now look at neuronal responses again. Not at attention, but neuronal responses, and you see that nicotine puts the receptor into desensitization. What it does to attention, and so we, of course, we inject also animals, and what it has also been tested in humans. The outcome usually is a bit confusing. So when you have smokers, you ask smokers to come into the psychology lab, and you ask them, of course, not to smoke on the morning. Then they perform, and they do an attention task. Then they perform pretty bad. But then when they can smoke a cigarette again, then after that they perform much better. So in that sense, you would conclude nicotine is increasing attention. If you ask non-smokers to come into the lab and they do the attention test, they have a very good attention, and then they smoke, then it usually either it does nothing or it reduces attention because they get a bit nauseous from smoking, a bit dizzy, and so in that sense, and the same you see with animals, with laboratory animals. So if you have an animal that's used to self-administer nicotine, then nicotine has a beneficial effect on attention. If you take a naive animal and inject it with nicotine, it usually reduces attention performance.
So does it have anything to do with nicotic acid, like in B3, if you take it orally?
I actually don't know, because of course in humans you because cigarette smoke contains thousands of compounds. So it's most likely it's nicotine, because in the laboratory animals it also is nicotine that does it. But it's a good point that it's hard to get a clean experiment to pinpoint that. When we look at the other type of receptors, the alpha-7, the fast responses, then we see that they hardly desensitize over this time. Hardly desensitize. So the beta-2 containing receptors, they desensitize very much. Alpha-7 not. So of course the next question would be, so if you have this network in the prefrontal cortex that expresses nicotinic receptors differentially, what would the presence of nicotine do to this network? So we started imaging again. Now we included a 10-minute application of 300 nanomolar nicotine and then applied acetylcholine again. And what we see is that, so here what you see is the control conditions. This plot you've already seen. So in the deep layers you see the most activation. But now when we pre-apply nicotine, then you see that in layer 2-3 there's hardly any activation anymore by acetylcholine. Nicotine pushed the receptors mostly into a desensitized state. In the deeper layers, let's say layer 5, you also hardly get any activation anymore, but in layer 6 there's still some activation left, which suggests that in the presence of nicotine, so in a smoker's brain, in the prefrontal cortex, only in the deep layers, functional acetylcholine receptors, nicotinic acetylcholine receptors are left over compared to the pre-nicotine condition.
Do you use acetylcholine esters inhibitors for this?
No. So to conclude the nicotinic part of the talk, after the next part is about the human brain, that's pretty short. But to conclude this part of the story, what we find is that nicotinic receptors are distributed over the prefrontal cortex network. Pyramidal neurons in deep layers express more nicotinic receptors that are also give much more depolarization, much more activation, and thereby these nicotinic receptors, when they're activated, they interfere with communication and rules for synaptic plasticity in the prefrontal cortex network. Also, nicotinic receptors are sufficient in the prefrontal cortex, are necessary and sufficient for proper attention function, and in the smoking brain, let's say, fast acetylcholine signaling through nicotinic receptors is limited to the deep layers, because in the other layers there's hardly any activation by acetylcholine left through nicotinic receptors. So, of course, we talk about these kind of mechanisms. I've tried to show you some relevance to mouse behavior, to attention behavior of nicotinic receptors in the prefrontal cortex. But of course, the question is, how do any of these mechanisms play a role in the human brain? So I think it's almost 2 years ago now that Guilherme started to collect human tissue from the operating room. The neurosurgeon operates on patients that either have untreatable pharmacoresistant epilepsy or have brain tumors. And to get to the epileptic focus or to the tumor, he needs to cut through cortex and sometimes cut through the hippocampus to get to the side, and then he removes that part of the tissue, and Guilherme takes it to the lab, and he makes slices of it. We record from neurons, individual neurons in the slice, and we load the cells with the biocyclic for reconstruction later on. And of course, one of the first questions that we addressed is, do human synapses show synaptic plasticity? There have been some studies done on synaptic plasticity with field recordings in hippocampus that show that you can get field LPP, but spike time independent plasticity was never addressed in the human brain, so we didn't know whether human synapses would be able to change strength in response to timed pre and post synaptic activity. So that's what we did, of course, time pre and post synaptic activity. Of course, you all know this curve for a rodent hippocampal culture. When you time, there are different small time windows in which the strength changes. You get timing LTP and timing LTD. So what are the rules for spike time independent plasticity in the human cortex? So we report from these human neurons, pyramidal neurons most of the time, and stimulate extracellularly.
From which part of cortex?
Temporal cortex. So always goes in from the side to dig to the hippocampus. So, of course, first we tried this small positive window where, of course, we expected to see LTP, and indeed we see very robust increase in the EPSP slope, so in the synapse strength. Then the next question, of course, was, well, when you take negative time windows, do you also see LTD? So we did minus 14 milliseconds, and to our surprise, we saw timing LTP at minus 14. Okay, so maybe human synapses can't show LTD. Oh, actually, so we did this. So this is what these curves are for the cortical synapses. The hippocampus part, Guillermo published earlier this year in one of the Frontiers journals. But the question, of course, is, do neocortical synapses also show these kind of windows? And what we wanted to know is, can you get synaptic repression? So when we increase the positive timing window, so presynaptic comes before postsynaptic, then what we observed is that we got timing LTD. So when you plot all these points of all these terms that we did into the classical curve, let's say, try to make the classical curve, then what you see is that in the negative time windows you get an increase in synapse strength, whereas in positive time windows you get more LTD compared to LTP. Okay, so we have a wide window in which you get TLTP, and in a positive window where you get TLTD. So somehow these rules for plasticity, I mean, of course the timing still matters, because if you go outside these windows, then you don't get strength, increases in strength or decreases in strength. So there is a temporal window in which you get plasticity, but apparently the rules for that are may differ. But of course, that's actually the question. The next question: is this timing window unique to the human neocortical synapses? So what we did, as you remember, in the rodent, we think of a window that's very different. So what we did is we compared the human data and to rat data, to adult rat data, and also temporal cortex, because that has never been done. So first of all these experiments were done with single exponential pairing. So another notion that's out there in the literature is that in adult cortical tissue you cannot get spike timing plasticity, spike timing LTP. So with a single postsynaptic exopotential. So that was the first thing that we did in the rodents with single exopotentials. In the temporal cortex in adult rodents, you also get a very wide temporal window for LTP. So you can get LTP, apparently, maybe it's dependent on brain area. In the hippocampus we also have data, small amount of data, single exopotential pairing, and you get LTP change in the strengthening of the synapses.
So the difference between this and the classical one is the age?
Yeah. And we did temporal cortex. People didn't record the temporal cortex in the rodents.
So it is not the age, even if you do it with juvenile.
Even if you would get
rats.
We didn't do juvenile rats temporal cortex, but so the difference between, you mean the difference between this and the literature, or this and
the B and Boo.
Right. Oh yeah. So B and Boo was cultures, hippocampal cultures, so taken from very young P0, P1 brain, and then put into culture for a couple of weeks, and then recording from pairs in cultures. So we do slices from adult rats. That's a big difference, yes.
But having only one pairing is
That's the question.
So what I mean is, with a single exopotential pairing, is of course we repeat the pairing, but it's not with one presynaptic stimulated with 5 postsynaptic exopotentials, but with 1 postsynaptic exopotential. So the notion in the literature is that in adult tissue you always need much more postsynaptic stimulation, because there's much more inhibition impinging on the dendrite.
But the question is still I think, is that a property of the temporal cortex, that it reverses the order independent of age? Is it that a property of a different cortex, or is it the property of an age? If you take, let's say, another cortex of human, I don't know what, the motor cortex, will you get also this, or is it specifically to the temporal cortex? Is this reverse?
So the reverse thing, so first of all, here you see hardly any LTD at positive. You see more LTP at positive intervals. You see some LTD, but they are in the minority. The majority shows LTP. And here we've quantified this. Let me show you that. So when you quantify minus 30 to plus 7, the boxes don't correspond, I see now. When you quantify plus 7 to plus 40, you see that you just average all the points. Here you see that in the humans you get a small LTD, whereas in the rodents you get an increase in synapse strength. And in the other windows, from minus 30 to plus 7, you get the LTP. So apparently this wide window for LTP, that's not unique to humans. That's also happening in adult rodents, apparently. Whether there's LTD, so there was also in literature, in adult rodent brain, it's very hard to get LTD. We see basically the same thing. It's hard to get LTD in the adult rodent. So this most likely that is opposite. Yeah, somehow that is unique to the human temporal cortex. I don't know whether other primates show the same thing, but.
You have data only on temporal cortex for humans?
And hippocampus. Humans, I don't show the data.
Hippocampus is the same.
Hippocampus, we also see a wide window. I don't have the data. Then we see that at this window, we also get LTD. So then it dips down below the curve, below the 0 line.
And you have LTD in the positive part?
So we didn't, in the hippocampus, we have much less data in the hippocampus because hippocampus we get much less than neocortex. So we didn't test this window here in the hippocampus.
What happened in red hippocampus with ADF?
So that's in the literature. What you get is a very narrow window and no LTD.
If you have more than one postsynaptic pulses like 5, as you do in the classical sense, does this change?
If we would do it in the human, where we do 5. In the hippocampus we did 2 actually potentials. There we showed that there was this white window that we found. But we didn't test with 5 here. With 1. Okay, so what are I should finish up. What are the mechanisms involved? So I can quickly go through this. We did NMDA receptors. Of course, they block plasticity. Calcium channels, we tested whether alpha calcium channels are involved. So we first applied nifedipine. They apparently are necessary also. You don't get much LTP when alpha calcium channels are blocked. And what we did is we applied BK. We made double recordings, not from connected cells, but from 2 neighboring cells. One with BK, which is basically an agonist of calcium channels. When the calcium channel is open, it prolongs the open time of the calcium channel. And there what we see is that it increases LTP at these negative intervals. So both in rat and in human, with BK you can increase the amount of synaptic gain at these very negative intervals. So the last data that I'll show you is what we think is that the secret of this wide temporal window lies in this strong after depolarization. So what you see is that this is 50 milliseconds. Both in human temporal cortex as well as in rat temporal cortex, you have this very prominent after depolarization. And of course that may be a reflection of calcium, dendritic calcium spikes. We don't know, we didn't record from the human dendrites yet, but that's the next thing that we will be after is to test whether this after depolarization is responsible for this wide window. When the EPSP rides on this ADP, then you get increases. When it follows after this ADP, then you get either no change or in the hippocampus you get a decrease. So that's what we think that is going on. Okay, so adult human and rat synapses show wide temporal window. I've shown you that human and rat synapses show TLTP at negative spike timings. In human synapses you get LTD at positive spike timings, and you need NMDA channels and L-type calcium channels to get plasticity, and these L-type channels may be playing a role in this setting this wide temporal window. Of course, I did not do this work alone. I didn't even do the experiments. All the people in the lab did the experiments, of course. There are a couple of people that I want to highlight. Rogier and Bernard and Karin took care of the nicotinic project. Karin is a postdoc that did all the behavioral day work and the virus work. Christian de Kok does a lot of the reconstructions in the lab. Guillermo, of course, you know, and Thijs, and I've got one picture of Natalia Borinova. These three people, they did all the plasticity work in the human brain. Thank you for your attention.